here in the last session I said this is an animal cell because we are finding a big nucleus at the center. Okay, then from the nuclear membrane, if you assume that this is the nuclear membrane, the membrane around the nucleus, from the nuclear membrane you will have endoplasmic reticulum, which may extend towards the plasma membrane like this. That's what so far we have seen regarding the endoplasmic reticulum. Now, the endoplasmic reticulum will be in continuation with another cell organelle called Golgi complex. Some will pronounce as Golgi.
Ma'am, how will you pronounce it as Golgi?
Golgi, Golgi, or Golgi. Anything is
okay. Doesn't sound good if you pronounce it as Golgi. It sounds like a 3-year-old is pronouncing it.
No, see, nouns we can pronounce it according to our slang. Nouns doesn't have any rules for accent. Okay, so this being noun, your comfortability. Golgi or Golgi. Golgi complex is also called as Golgi organelle or Golgi organelle. Okay. So organelle, we already discussed, cell organelle always stands for living part of the cell, isn't it? Cell organelle. If this is the living part of the cell, it should definitely carry a function, isn't it? So when you take the Golgi complex or Golgi complex, it will be in continuation with the endoplasmic reticulum. See, if this is the endoplasmic reticulum, it is in continuation with the endoplasmic reticulum. Why there is a continuation between endoplasmic reticulum and Golgi complex? That's the point we'll discuss over here. Generally, Golgi complex is called as traffic police of the cell. I hope this is a terrible word you would have heard. Traffic police. Traffic police, what they'll do?
They will check correct on the signals like that. The maintenance of order.
Control traffic. So indirectly, Golgi apparatus are like it checks what is coming in and what is coming out.
Yes. Yes. Good response. Yeah. Traffic control. Traffic control means like the other student said, controlling traffic based on the signal light, something like that. So with the signal, traffic will be controlled by a traffic police. Okay. So here Golgi complex, when we are saying they act as traffic police of the cell, so they should get a signal, isn't it? What is the signal over here for Golgi complex? The signal for Golgi complex is the protein. So where are
protein synthesis by ER?
Yeah, where are proteins are getting synthesized? They are getting synthesized in endoplasmic reticulum. While discussing about endoplasmic reticulum, what we said, from the endoplasmic reticulum in the form of vesicles. You remember this word, vesicles? From here, after ribosomes, the vesicles will get pinched off. They'll come out. The vesicles will come out. So these vesicles are with the proteins. Okay. So the vesicles with proteins will come out from the endoplasmic reticulum, which triggers the signal for the Golgi complex to receive the proteins. This is what makes the Golgi complex a traffic police. It controls the movement of proteins. The protein movement is controlled. How the protein movement is controlled by the Golgi complex? For that, we have to understand the structure of Golgi complex. Golgi complex structure possesses two phases. One phase is called cis phase. The other phase is called trans phase. Cis phase, trans phase. So what does mean by cis phase and trans phase? Suppose we are getting the proteins from here, from the endoplasmic reticulum. The receiving end of the proteins from the endoplasmic reticulum, this part, okay, that receiving part of the proteins is called cis phase of Golgi complex. Once they get the proteins will be packed. We'll see this word in functional aspect of Golgi complex. For time being, just understand the received proteins from the cis phase will be packed by the Golgi complex and they will be sent to destined part through the trans phase. It's like the cis phase is the receiving end and trans phase is the transport end. Got the point? The cis phase is the side of the Golgi complex which will receive proteins from the endoplasmic reticulum, like this part. And inside the Golgi complex, they will be packed. The proteins will be packed. And from the other end of the Golgi complex, this end is called trans phase. From that phase of the Golgi complex, the proteins will be transported. Usually the cis phase and the trans phase do carry other names also. Cis phase is generally called the convex side of the Golgi complex. Convex side. Trans phase is usually called the concave side of the Golgi complex. Okay, so the structure of Golgi complex, if you take, the Golgi complex acts as the traffic police by getting signal from the endoplasmic reticulum in the form of proteins. Proteins will enter the Golgi complex cis phase and after getting packed in the Golgi complex, they will move to the trans phase and get transported. Okay. Show us, take this as the Golgi complex. The whole Golgi complex looks like this. Okay. Now here, the vesicles move from endoplasmic reticulum to Golgi. Yes. Vesicles are with proteins. They are produced in endoplasmic reticulum. Then from endoplasmic reticulum, it is in continuation with endoplasmic reticulum. Here you will find the endoplasmic reticulum. From the endoplasmic reticulum, the produced proteins in the form of vesicles will come and get into the cis phase or the receiving side of the Golgi apparatus or the Golgi body. Here they used the word coalesce. See the word coalesce. Coalesce means what? They'll get clubbed. They'll get moved into the vesicles with proteins. They'll move into the cis phase and through the Golgi apparatus, they'll move towards the trans phase. Trans phase, trans side. Okay, that's called a shipping side or the transport side. Shipping means what? From that point, it will get transported to another point. That's called shipping, isn't it? In usual common language. So here, this is the end which looks like concave when compared to this end which looks like convex. So you might be familiar with convex and concave sides, physics. So the convex side is the cis phase, which receives the proteins. They'll move through the Golgi complex and they will be coming towards that trans phase also called as transport phase, and they will move from here to the destination part. Within the Golgi complex, various functions will happen. What different types of functions will happen, that we'll see in the next part of the slide. Is this structural aspect clear? Saying that the Golgi apparatus will have a cis phase, trans phase, and in between it's showing some packaging of the protein. Okay, so what does mean by that packaging, we'll see here. So like if you remember, the Golgi complex is in association with endoplasmic reticulum point. While discussing the endoplasmic reticulum also, we talked about structures like cisternae, plural word. Okay. Then tubules
And vesicles.
Yes, vesicles. All these three parts, cisternae, tubules, and vesicles, will find in Golgi apparatus also. The same structural aspect, if you just compare the endoplasmic reticulum structure with the Golgi complex, see here, these are nothing but the cisternae. What I told about cisternae, the cisternae are sac-like structures, flattened sac-like structures. So this is what the cisternae are. This part. Okay. Then we have tubules. Tubules means there will be tube-like structures which are branched or unbranched with one another. Something like this. This part are tubules. Then from the tubules we will get some rounded structures moving out of the cisternae and tubules. These are called vesicles. Cisternae, tubules, and vesicles. All these will find in endoplasmic reticulum as well as in Golgi complex. Okay, because both the apparatus are dealing with proteins. That's why we find almost similar structure. We'll find one more extra structure in Golgi apparatus, that is called Golgian vacuole. Golgian vacuole. This is the fourth structure you will find only in Golgi complex. So that's the structural difference between the endoplasmic reticulum and Golgi complex. We compare the structural difference between endoplasmic reticulum and Golgi complex. Only this Golgi and vesicles are absent in endoplasmic reticulum, and we'll find them only in Golgi apparatus. Okay. Yes.
I didn't understand Golgi and vacuole.
Golgian vacuoles are the one which will get pinched off. These vesicles are formed with proteins carrying from the endoplasmic reticulum. They'll get pinched off. Pinch off means what? They'll get removed from the structure. Know the word pinch off?
Yes, ma'am.
They'll get removed from the structure of Golgi apparatus. They'll come freely outwards, and they have to be transported outside to the destination. Now, like this. Shipping here, what we used near the transphase. That's what these things will get shipped out. The Golgian vacuoles with proteins.
Ma'am,
Clear?
is the endoplasmic reticulum vacuole will get transferred into Golgi and vacuole, is it?
No, endoplasmic reticulum, it will transfer vesicles, not vacuoles. Because they're not packed properly. They're just produced. That's why they're coming in a unmodified form from endoplasmic reticulum to Golgi apparatus.
Yes. Yes.
Yes, you got the point.
Yes, ma'am.
Why the vesicle word is used over there? Because in endoplasmic reticulum, vesicles will be with proteins produced there.
Yes, ma'am.
And Golgi apparatus, further processing and packaging happens to the proteins. And that packaged proteins will be moving outside of the Golgi The other word in some books they use post office of the cell. That we'll see why they are called post office of the cell in function. These are the synonymous words you can say for Golgi apparatus. Okay. So the Golgi apparatus major important point is the cis phase. The word phase, cis phase, then trans phase. These are the two important aspects related to structure of Golgi apparatus.